Hey guys, Mr. Hyatt here. This is part 3 of the meiosis lecture. So, we looked at a little bit of math in part 2. We said there are 8,388,608 different versions of sperm cells that I can create, and the same number of eggs that my wife can create. Put those together, that means that there are over 64 million combinations of my chromosomes and my wife's chromosomes. That kind of makes me feel, I don't know, a little bit special, like I'm one of 64 million possibilities that I could have been. It kind of makes you feel, I don't know, small when you see a number like that. So think about what that means in terms of your siblings. I mean, we look a little bit like each other, but we all look different, unless you're an identical twin. It's crazy that there are 64 million combinations that you could be, and that resets every time. So it's no wonder that we have some traits in common, but overall we look different. The thing that really throws a wrench into all of this math is crossing over, and makes this mathematically impossible to calculate. Those face combining apps and things like that you see sometimes on Facebook are just ridiculous, because number one, they can't account for the 64 million combinations that random fertilization ensures and independent assortment ensures, but they definitely can't quantify crossing over. We'll do a lab that'll modify or will model crossing over. That I think will help it make a lot of sense. But it's where we're going to have trade material. These chiasmata are points of contact. We see these chromosomes where they overlap. And I like to explain it like when you were a little kid and you fell asleep chewing gum. It's a lot easier for your mom to just cut the tangle out of your hair than it is to try to untangle hair by hair. And that's the same logic that our cells are going to use. So we see these chiasmata where we get some crossing over and we swap some DNA, so we end up with an all-blue chromosome, a chromosome that's mostly blue with a little bit of red, a chromosome that's mostly red with a little bit of blue, and then an all-red chromosome. So now we've got even more possibilities for chromosomes, especially combinations. It's going to break old linkage groups for sure. It's going to create new linkage groups that are going to increase variation. Really common during meiosis. Think about it though. It happens in prophase. Prophase 1 is when we've got essentially 4 copies of every chromosome. And they're all going to be piled up. They're going to stick to each other. They're going to be very sticky with one another. And so that makes sense that that's when it would happen. Notice that we can use the frequency to map the positions of genes on chromosomes. So think of it this way. When you were a little kid, you probably played Red Rover. If you are on the far left end and your best friend is on the far right end, you're probably going to get split up a bunch of times. That means you're not very close together because you've been split up so many times. If you and your best friend are side by side, you're not going to be split as many times. Every person that runs from the other side to your side isn't going to split you up. So you're going to be split up far less often, which implies that you're closer together, if you don't know your original position. I think that makes sense. So with crossing over, an offspring can never be 100% like a parent if we're using sexual reproduction. Because some of my chromosomes that I passed on to my son are partially from my dad and are partially, well, let me say that a different way. Some of them are even partially from my grandpa and partially from my grandma, because my dad's chromosomes crossed over in his prophase. So then I inherit a chromosome 1 that's partially my grandma, partially my grandpa, and then it crosses over with one that's from my mom's side, and now we've got all sorts of variation. Multiple crossovers are really common on large chromosomes. You see kind of what that would look like here. Smaller chromosomes, obviously we've got smaller surface area. Genes near the centromere aren't going to cross over very often because they're tightly wound to the centromere. So just to summarize these 3 presentations, make sure you know how the chromosomes separate during meiosis. Make sure you have a mental image in your head of how those chromosomes move around. Know the terms, what we call them at each different phase. Are they a tetrad? Are they a homolog? It takes a little bit of practice with them, but you guys are all smart people or you wouldn't be here. Know the comparison between meiosis and mitosis. Be able to explain how each of those 3 things we talked about increases variation through sexual reproduction. That's meiosis.